thank you for having me here to talk today about some of the work we've been doing. I'm Soumya Raychaudhuri, and as Carlos mentioned, I'm at Brigham and Women's Hospital, where I practice as a rheumatologist, and I do bioinformatics and computational biology in our lab, which is at the Broad and at Harvard. So let's see. So the main disease I'm going to talk about today is rheumatoid arthritis. So for those of you who don't know, I suspect many of you are already familiar with rheumatoid arthritis, but rheumatoid arthritis is a disease where your immune system attacks the joints, and typically it attacks this very specific tissue called the synovial tissue of the joints. It's this thin capsule surrounding the joint space. In some sense, it's a pretty new disease. It was only really defined in 1800 by a student in their dissertation. The student called it asthenic gout. I think before 1800, any type of joint inflammation was just called gout. But this student recognized that this form of gout occurred more often in women. They had chronic course. So it wasn't sort of a flaring joint pain that got better, but it was sort of persisted. And it was polyarticular. It affected many joints. And this is Pierre-Auguste Renoir. Renoir was affected with rheumatoid arthritis, and in the late stages of his life, his hands were so badly affected that paintings like this, they were done with a brush strapped to his wrist. So this disease is one that affects about 1% of the population worldwide. It leads to symmetric destruction of joints, particularly in the hands. Other joints that can be affected, such as the ankles, wrists, neck, etc. And it's mediated, as I had mentioned, by immune infiltration of the synovium. So if you have this sort of thin, can you guys see my mouse? No? Yeah?
Yes.
Okay, perfect. So you can see there's this thin layer called the synovium, and that's what's getting inflamed when the immune system gets in there, causing hypertrophy of that tissue, and that tissue then goes on to destroy the joint, and that leads to disability. It's characterized by autoantibodies, antibodies made by B cells. It can have systemic complications. I'll give you, just to illustrate some of the challenges around rheumatoid arthritis therapy, thought I would tell you about actually one of my patients. This is a 59-year-old attorney who I see in the clinic. She was a patient I met in July 2018. And she, at that time, had 6 to 8 months of hand stiffness. And the stiffness and swelling in her hands made it difficult actually to do her work. She was put on ibuprofen or Advil, and then eventually put on something similar but a little stronger called meloxicam. She socially lived with her husband and her cats. She worked at a law firm. She had grown kids. Smoking is an important risk factor for rheumatoid arthritis. She quit smoking in her 20s, so probably not an issue for her. And her mother apparently had a history of rheumatoid arthritis, which is another point about these diseases. Autoimmune diseases like rheumatoid arthritis are often familial. On the physical exam, her right index proximal interphalangeal joint, which I'll show you a picture of it, had swelling. So she had a swollen, basically a swollen index finger. When you squeezed her hands, it hurt. Her rings looked tight on her fingers. And we stopped the ibuprofen-like medication, the meloxicam, and she got worse. So these were some of her labs. The antibody labs we normally use for rheumatoid arthritis, CCP and rheumatoid factor, they were negative. And in about 30 to 40% of patients, these antibody factors that we like are often negative. ANA, which is an antibody for lupus, but often positive in individuals with rheumatoid arthritis, was positive for her. We did a bunch of other tests that were negative. And this is her thumb. And you can see this is like not normal, right? This is very swollen. And you might understand why this could be painful. So first I put her on a medication called Plaquenil or hydroxychloroquine. medication, which is a kind of a mild anti-inflammatory. It enjoyed a little bit of fame during the COVID pandemic, but not very effective for COVID, but a little bit effective for rheumatoid arthritis. She actually had an allergic reaction to the Plaquenil, had facial swelling, and after a little bit of back and forth, we decided it was an allergy. So her joints, after we stopped it, of course, a month later got worse. So we put her on steroids briefly, and she got a lot better. Then the pathway I'm about to show you, I mean, if you pay attention to the amount of time passing, it's really notable. We put her on a medication called Arava, which is an mild immunosuppressive. She got a little bit better, but with the Arava, she had a side effect. She had hair loss. So then we thought about methotrexate. Methotrexate start was delayed by about a year due to the COVID pandemic. But then she didn't really respond to the methotrexate, so we put her back on steroids. We don't love to have our patients on steroids because it creates so many problems. And then finally, we put her on a biologic, Humira, and she's under reasonable control, but it definitely isn't perfect. So the takeaways, so her case illustrates some of the sort of typical things of our patients with rheumatoid arthritis. One is, the serologies, the antibodies, which we think are so useful for rheumatoid arthritis, and they really are, can be very atypical. They're not, there's heterogeneity. Some patients have them, some patients don't. There's a lot of great medications available. We tried her on a bunch of them, but it can take years to find the right medication for the right patient. And in those years, patients often have joint damage, which is permanent. And even now, my patient continues to have some pain despite being on Humira. So it's hard to know if she's on the best possible therapy or if we should be trying something else. And if you're not treating your patient perfectly, there's joint damage, quality of life issues. There's actually an increase in cardiovascular risk because the inflammation puts them at risk of having a heart attack. And obviously, we don't like to have our patients on steroids. In terms of pharmaceutical development, 1998 is when things really started to change in this field. You can see that there's a whole bunch of biologic medications that started emerging around that time. Adalimumab or Humira is what we've been talking about here, but now you can see there's lots of different pathways we can target with these different medications, including the JAK-STAT immune pathway, including the TNF pathway with the anti-TNF agents, and so on. But despite the improvement in therapies, there's still gaps. There's still patients with refractory rheumatoid arthritis, and there's a real need to get our patients to the right treatment quickly, and immune suppression, which we love to use, doesn't always consistently lead to remission, so there's still no cure. So today I'm going to talk about two different approaches we've been taking to really understand how to treat a disease like rheumatoid arthritis effectively. There's one approach is investigating human tissue inflammatory architecture. So defining key pathogenic cell states in disease. So can we use single cell technologies and other related technologies to figure out what the key cell states are? And maybe that leads us to possible pathways we can target. Also, we can think about human genetics, which is very useful in that it defines causal pathways, but defining the function of those genes is often hard. Both require careful and rigorous computation and statistics. So again, as I mentioned, that we're going to be thinking a lot about the tissue, that synovial tissue, which, as you can see in patients with rheumatoid arthritis on the right, you're looking at the top of a knee here. You can see that synovial tissue, barely visible on the left, is inflamed on the right. So we're going to talk about getting biopsies and looking at single cell data there. Also, in terms of thinking about the genetics, rheumatoid arthritis is familial. So it's present in about 0.5% of the general population. But if you have an affected first-degree relative, your risk goes up about 4-fold to about 2%. And this is data from the Swedish population registries. Indeed, most of the autoimmune diseases do have an increased risk if you have an affected first-degree relative. You can see that the numbers range from sort of 5 to 50, which is tells you that it's a complex disease. So it's not a Mendelian disease where these numbers would be off the scale in the hundreds, thousands range. But it's also not like it's not genetic, in which case these numbers would be much lower. So this suggests that there's going to be many alleles, each of which will have a weak effect and interact with environment in some sort of way. So the tissue is the issue. So identifying key cell states in the context of rheumatoid arthritis tissue inflammation is going to be one of our goals. The other one of our goals is using genetics to get to causal mechanisms. So it's really great to find a cell state that's expanded in RA cases, but how do you know that it's not a passenger population? How do you know it's actually driving inflammation? And I'm going to argue that this is where genetics can be really useful as we try to understand what's going on. The first part of the talk, I'm going to talk about work we've been doing to define key cell states in tissue inflammation. Indeed, if you look at this inflamed synovium, you find all kinds of different cell types. What are the key cell states? Which ones are expanded, and so on. I'm going to mention that a lot of the work I'm presenting here is part of a large consortium, which I've had the pleasure to be a part of. It's called the AMP, Accelerating Medicines Partnership Consortium. It's a private-public partnership, NIH, working with a lot of pharma partners. And there's definitely a lot of people to thank, both on the private side and on the public side. And I want to especially acknowledge these individuals who've been really driving the project: Fan, Helena, Parna, Kevin, and Mia, who are rising scientists who did a lot of the work I'm going to present. So we've known from studies that we've done in the past few years that there are key cell populations that are playing a role in rheumatoid arthritis. CD8 positive, granzyme K positive T cells are seen in tissue inflammation. T peripheral helper cells are a population we described with Deepak Rao and Michael Brenner. Autoimmune associated B cells, another population that we've seen to be associated with RA inflammatory fibroblasts, MERTK positive myeloid cells. So the question we want to ask is there a structure to synovial tissue inflammation? So are these cell states always present? Are they always expanded in all patients with RA? Or are there tissue inflammatory phenotypes that are enriched for certain of these cell states and depleted for others? So can we kind of deconstruct disease is what we're trying to do here. With the long-term thinking that if we found these different tissue inflammatory architectures, maybe they'd respond better to some treatments and not others. Of course, the work I'm going to show has really greatly benefited from this explosion in single cell technologies. This project really started right around when the 10x technologies came online. So that really allowed us to look at thousands of cells from 80 individuals, large number of individuals, and gave us a lot of power to do these studies. And for those of you that are a little bit unfamiliar with the single cell field, one of the approaches we were using was a method called REAP-seq or site-seq or total-seq. It has a bunch of different names. Bottom line is you have an antibody surface marker staining that you can do. So these antibodies are labeled with oligonucleotides. And normally we do FACS analysis, and FACS allows us to say which cells are expressing CD4 protein or not, or CD8 protein or not, or some of these key populations. Site-seq allows us to do that in a sequencing based approach. So it's very important for a lot of these immune populations where our understanding is very much driven by surface marker and FACS. So this is a pipeline. So all of these samples were obtained in clinical sites, these synovial biopsies. These biopsies were obtained and shipped to Boston, to Michael Brenner and his team, Helena Johnson and Kevin Way, where these samples were then disaggregated and sent for multiple technologies. I'm going to be focusing a lot on the 10x technology, where they were assayed with site-seq to obtain RNA and protein level data. I won't get into too much detail here, but basically the patient cohort was really diverse. So we had individuals that had not been treated with any drugs. That was about a third of the individuals. We had individuals that were treated with methotrexate, which is the first drug we use in rheumatoid arthritis, and they hadn't responded. And we had another third of individuals that had been treated with an advanced biologic. And these are individuals that hadn't responded. So these were all patients that had like high disease activity, basically. But they were fairly diverse in terms of their treatment history, because we wanted to make sure we're capturing a pretty good cross-section of what RA looks like out in the wild. So doing kind of some standard single cell stuff, we analyzed our data on 328,000 cells from 80 individuals. And we identified the major populations here. So T cells, NK cells, myeloid cells, stromal cells, endothelial cells, and B cells. Those were the main populations. And you can see that on the left, we've clustered on RNA. And because we have the protein data, we can make sure that the populations are positive for the key protein markers. One of the things we saw immediately is that there are these like, so on the left, you see there's osteoarthritis. On the right, you see our RA patients. You see this tremendous heterogeneity. There are some patients that have a lot of T cells in the purple, some patients that have a lot of endothelial cells in the blue, some patients that have a lot of fibroblasts. So the major cell types are like different from patient to patient, and we can categorize them, and we've categorized them here and name those categories as F or EFM or T plus B and so on. So are there clinical implications for this group? So that was our first question. Are these patients all the same, or if you have these different phenotypes, might you be different? So the first thing we did was we looked at histology, and we looked at basically several histological markers. One is density, which is like how much is the density of your immune cells, and you can see that this group, patients are different. There's significant differences there. We can also look at aggregates. So are we seeing sort of clusters of immune cells? And again, you can see that that's different across the different groups. And then finally, you can look at autoantibodies, and you see that that's different too. So the individuals with lots of T cells and B cells, very important for making autoantibodies. They're the ones that have the highest autoantibody titer. But super interestingly, if you look at these other two metrics, DAS28 or CDAI, these are metrics of like how active is a patient's disease. And you can see that this is not different. So basically, there's patients with active joint disease, patients that are going to be needing therapy regardless of what group you're at. So it's not simply that we're finding people who are very active and people who are unactive. One really important question was treatment response. So these CTAPs, they're defined from single cell data, but they basically represent major cell types. So we realized that we could look at bulk RNA-seq data and deconvolute bulk RNA-seq data and basically estimate from synovial tissues with bulk RNA-seq data what CTAP we think they might be. So, and we took advantage of some public clinical trial data where bulk RNA-seq data was obtained. And this was really interesting. We looked at treatment response, and this to me suggests that these CTAPs could be informative. What you see is that individuals who have this F CTAP phenotype, they're very resistant to treatment. So these are people who have a lot of fibroblasts, inflammatory fibroblasts, and our guess is most of our therapies are working on T cells, B cells, et cetera. So if you have a lot of fibroblasts, you're unlikely to respond to therapy. So again, suggesting that there's a kind of an unmet need there. Are there different cell states dominating these different CTAPs? So the next question is, if you're in CTAP TB with lots of T cells and B cells, or you're in CTAP TF with lots of T cells and fibroblasts, are they the same T cells or different T cell states? Because again, that may have therapeutic implications. So here are 6 major cell types. We can cluster them into these very fine populations. I'm going to zoom in on T cells and look at this analysis here. And what you see is that we have, 24 different T cell populations as indicated here. We've gone through some pains to annotate them. Just give you a little bit of a flavor of this. You can see the left and right of this butterfly reflect CD4 staining versus not. So CD4 T cells versus CD8 T cells, the two different kinds of T cells. You can see that yeah, here we've got the CD8 T cells, and you can see there's a group of T cells that are CD25 positive. Those are your Tregs. So now we can go through and start annotating things. There's one cluster here, cluster 8. These are T peripheral helper cells, which are important in that they're expanded in RA. You see that these granzyme K positive CD8 T cells, they're here too. We see this population really enriched in inflammatory tissues. Yeah, cluster 3 and 7. These are the TPH cells. You can see that they're PD1. They're a PD1 positive population that's also negative for CCR5. T follicular helper cells are positive for CCR5. So some of the key populations that we expected to see, we did see. And then some simple question: are the T cells associated with specific CTAPs? So we see multiple T cells dominating the CTAPs. For example, if you look at T cells, there's some subset of patients with T cells and fibroblasts, some subset of patients with T cells and B cells, some subset of patients with T cells and myeloid cells. Are granzyme K T cells, TPH cells, are they enriched equally in all of them? So to this, we developed a method called covariant neighborhood analysis. One simple question we could have asked is, are these clusters up in some subset of patients? But we were worried that the clustering resolution isn't always ideal. Clustering obviously forces us to make some arbitrary choices about how big we want our groups of cells to be or not. And transcriptional heterogeneity that's important could be obscured by clustering. So our approach was this like neighborhood-based approach. So we developed a nice method a year or two ago, led by Kira Sheff and Lori Rumker, where what we did was we basically look at every cell, do a random walk to define neighborhoods of cells. And then we defined these 5 example neighborhoods as indicated on the right. And then we look for neighborhood covariance. So we look for samples where the same neighborhoods are up or down together. And to find that, we simply use principal components analysis, which helps us identify neighborhoods that are correlated across individuals. And then finally, we can test those principal components for association. So to give you a flavor of how this works, this is a natural killer cells. You can see there's 13 clusters here. You can test the clusters all individually. You start to see that there's some trends where like T plus F has this generally a little bit expanded expansion of NK of these top 3 populations. The other thing we can do is apply CNA. And when we apply that, we see really screening statistical significance. You see that there's some subset of cells that are expanded in T plus F patients, some other subset of NK cells that are depleted. Now we can say, well, what clusters do they represent? And what we see is they're largely CD16 positive clusters. So not just a single cluster, but an aggregation of them that are indeed expanded. And of course, we can now go and look at those cells, look at the genes, and what we see is that these are interferon gamma, responsive NK cells. So now what we can do is say, okay, what's the heterogeneity in RA? And now we can start filling out this kind of table where we say, okay, here's these particular different subcategories of tissue inflammation. T plus F, has expansion of some of these NK populations, depletions of others, right? Now going back to T cells, let's look at those individuals that have CTAP-TB. You see that there's some populations on the left that are really expanded. So these are your T peripheral helper cells, very important in RA, but they're very particularly expanded in individuals that have a lot of T cells and B cells, these CTAP-TB individuals. And again, so you can look at this by looking at these individual clusters, breaking down which cells are expanded versus not. And again, as I mentioned, this was a population we described a few years back in RA. And what we see indeed is people have more TPH cells. They're the individuals that have more autoimmune associated B cells. So that those two things go together. What about the people who have lots of T cells and fibroblasts? Do they have the same T cell expansion? No, they don't. Look at the right. you see that the red has moved to other areas. You're seeing an expansion of naive cells and also CD8 cytotoxic granzyme B positive TEMRA type cells. What about B cells? Same story. If you look at individuals who have CTAP-TB, they have a lot of B cells, but even amongst those B cells, the most expanded population are these populations here in the red. These represent the age or autoimmune associated B cell population. So again, now we can fill out this table a little bit more. And myeloid cells, again, very interesting story. Depending on what type of disease you have an expansion of different sorts of myeloid cells as indicated here. So again, we can fill out some more information. We can keep going. And for every cell type, we can see which cell states are relatively expanded in these different tissue phenotypes. So CTAPs favor actually different favor cell states. So it's not just that some of these CTAPs have more T cells and B cells, they have different types of T cells. You know, TF, CTAP, the T and fibroblast individuals, they have a lot of interferon gamma signal for T cells and NK cells. TB, CTAP, which is kind of maybe what we canonically think of as RA, has a lot of T peripheral helper cells and age-associated B cells. The MCTAP has a lot of inflammatory macrophages. So the people who have a lot of myeloid cells, they have a lot of macrophages that are inflammatory. And there's important therapeutic implications because existing therapies may be important for certain CTAPs. So anti-IL-1 approved for RA therapy doesn't work really well in most patients. Perhaps it's going to work well in CTAP-M because those individuals have a lot of these myeloid cells that secrete IL-1. And then there's a whole bunch of therapies that block T cell, B cell interactions. Maybe those therapies are going to work really well in people who have CTAP-TB. Just to summarize this section on the AMP, this was a collaboration of clinicians, lab-based researchers, and computational biologists to really understand the synovial architecture of disease. We defined these CTAPs, which are defined on the basis of common cell populations, and it's really easy to deploy in clinical practice. And then there's evidence of association of clinical outcomes. Each CTAP is associated with key functional states, which suggests there may be implications for using specific therapeutics in specific individuals. And the question, of course, is, are CTAPs genetically determined? Do they represent disease subphenotypes, or are they stable over time? Are they stable across strains? So now I'm going to switch gears. We've been talking about correlations of cell states with different phenotypes. What about the genetics? Are they going to help us identify the cell states that are maybe more causal and playing an important role in causing disease? How can these cell states help us understand genetic susceptibility? So can genetics help us identify new therapeutic target? Yes, there's strong evidence that risk genes are better targets than just other genes. Identification of cell states associated with disease or disease phenotypes can be informative. But in the work that I've been showing, as I've been saying, identifying cell states that are reactive versus driving inflammation, that's hard. But risk alleles and genes can help us point to causal cell states, and the alleles may be acting in T cell states. And the genes they may act on may be expressed in T cell states. So in terms of the genetics, you know, we have been privileged to be a part of a global consortium of individuals who've been trying to map risk alleles in RA, and these are my key collaborators who I've been working with in this space. And I also just want to acknowledge all our international collaborators. Obviously, studies like this require lots of coordination throughout the world. Samples here for this genetic study came from, Africa, the Middle East, South Asia, Europe, and East Asia, and included something like 100,000 cases and controls. And this study, one of the things I really like about it is we're looking at many different populations, which not only increases our power, but also helps us find alleles that have kind of consistent effects everywhere, which are probably the most useful in terms of mechanism. This is a principal components analysis plot and a UMAP of our samples. So you can see that based on genotype data alone, we're able to separate out the different populations, not surprisingly. And we found about 124 loci. When you combine that with the alleles that were known before, it's about 150. littering every region of the genome. And now what you can see is that with each year that's gone by, we found more and more and more of these alleles. T cell biology is strongly implicated by most of these alleles. The HLA locus is very important for RA and other autoimmune diseases. It's intrinsic to the interactions with T cells. But outside of the HLA, most of the alleles are non-coding, 90% of them, and they're disproportionately close to T cells, and they're disproportionately in enhancers and regulatory regions that are T cell specific. So in those regions of the genome, they're playing an important role in controlling T cell genes. And these are some of the key results, which I won't belabor. But the sort of typical mechanism that we imagine, so most of these alleles are SNPs. They're sitting in a promoter or an enhancer, a regulatory region. And we imagine that there's a regulatory protein, like a transcription factor, excuse me, which would normally bind here and turn on a gene. But now if you have a SNP, it's going to affect the efficiency with which that binding happens, and that's going to affect how well things go. What we'd expect to see for a lot of these situations is something like this, where you have a SNP, it's acting on a gene. And so the more copies of the risk allele you have, the more your expression is affected. So this is a SNP associated with RA, and you can see as you go from GG to CC, your expression levels go from high to medium to low. So we call this an expression quantitative trait loci, or an EQTL. So this is a great example, because here's the SNP that's associated with RA. And now when you condition on it, what you see is that all the association expression goes away. So the same SNP that's driving RA susceptibility is also driving association expression. So we would conclude that the SNP is working by altering the expression of this gene. Now, in practice, however, what we see is that most risk alleles do not alter gene expression. So about 50% of autoimmune risk alleles and rheumatoid arthritis risk alleles are in regulatory regions, T cell regulatory regions, but most aren't EQTLs. Most don't alter expression of T cells, and so what's kind of going on? What we think is probably happening is that functional states matter. So again, there's this wide diversity of T cell states that I showed you. Is it possible that the effect of a SNP on gene expression is state dependent? Perhaps, as T cells go from naive to memory to different types of memory T cell states, those important regulatory regions are only active in certain important states, but not in others. So the effect of the allele on gene expression happens sometimes, but not all the time. So we've spent a lot of time thinking about state-specific effects. So thinking about how alleles act in specific states where enhancers and regulatory elements are the most active. And of course, we would expect that these downstream genes that are important are going to be specifically expressed in pathogenic cell states that are important. So here you have a SNP, which, this is again what we're interested in. The SNP maybe doesn't affect expression in general, but maybe in some cells on the right, you see a correlation of the SNP affecting gene expression. And indeed, we see that there are these kind of dynamic EQTLs for some important genes like ORMDL3. What you see is like not so associated with gene expression in a lot of cell types, but in cytotoxic T cells, there's a strong association. One example I like a lot is CTLA4. CTLA4 is highly expressed in these T cells over here. These are regulatory T cells. And the association of SNP to expression, you can see the beta is really low. So the SNP isn't really affecting expression when the gene expression level is high. But when the gene expression level is low, as it is here, that's when the SNP really matters. So, what we're really seeing for some of these SNPs like this one is sort of, it's not affecting the expression when the expression is high, but it's actually affecting the expression more when you're not supposed to see expression. So it's kind of like an ectopic expression situation. Here's some interesting SNPs that came out of our genetic studies. I'm just going to talk about a few of them. LEF1, which I think is particularly interesting. This SNP that we find associated with RA, it sits right in an intron of LEF1, so we think LEF1 is likely to be a causal gene. And this is where it gets interesting. So if you look at our T cell states, LEF1 isn't expressed in all T cells, but it's expressed only in some. And you can see the pattern here of its expression. This is, again, that AMP data. Now look at that. When we asked which T cells are associated with the TFC tap, we see the states that are expanded with this particular phenotype, and the patterns really do match nicely. Here's another gene, JERD2. The SNP is an EQTL. The SNP affects the expression of this gene JERD2, so we think JERD2 is probably a causal gene. Look at its expression. It's expressed only in this upper left region here. Remember CTAP TB, TPH cells were expanded, so JERD2 expression is very specific to these TPH cells. Here's another example, WISP1. This is a stromal population. You see it expressed in fibroblasts, stromal cells. That's an interesting one. IL-6R, IL-6 is an important RA drug target, already known. It's expressed, you can see there's multiple SNPs that affect expression. They're independent of each other. So you can see there's a purple SNP and a blue SNP. The purple SNP is associated with expression of monocytes. Blue SNP is associated with expression in T cells. Let's look at monocytes. You see that IL-6 receptor is kind of expressed everywhere. Now this is a little more interesting. IL-6 receptor in T cells, you can again see there's high expression on the left side in CD4 T cells and the highest expression in TPH cells. Again, those are the populations expanded in a subset of individuals with RA. So summarizing the genetics, the genetic discovery of alleles outside of HLA has been very productive. Multi-ancestry studies are leading to rapid discoveries. Many of these alleles work by altering gene regulation. Defining the regulatory function of these alleles requires us to think specifically about cellular context and the function of alleles that change with cell state. The data on inflammatory tissue is key and reveals that key genes are expressed in very specific cell states. It argues that these cell states are important to RA mechanisms. And these studies are pointing us to causal genetic mechanisms in RA, causal cell states. So to conclude, we think there's an amazing opportunity to think about sub-phenotypic disease using functional genomic and single cell data. This work is now moving on to starting to think about spatial transcriptomic data. We've demonstrated this potential in rheumatoid arthritis. And it really does require us to think about tissue and tissue architecture. Finding an associated cell state is not the same as linking it to mechanism. Genetics here can be helpful because genetics helps us find the genes that are driving disease. If we see which cell states they're acting in, which cell states they're expressed in, which cell states they have a function in, that tells us that those cell states are maybe useful cell states to think about in terms of targeting and therapeutic development. Finally, I just want to thank Carlos for having me here. As Carlos mentioned, he was an instrumental person in my career when I was just getting started as a computational biologist back in 1995. Carlos, it was a little while ago, pre-Human Genome Project time frame. And finally, I just want to thank our funding agencies, and of course the lab. We're here on one of the Harbor Islands enjoying our retreat here. So with that, I can stop and I can take some questions. Thank you.
Awesome. Thank you so much. It's a great pleasure to have you here. And let's see if we have any questions.
There's actually a question in the chat box. Maybe.
Yes. Yes.
Brian, this is your question. Do you want to read it, or I'm happy to read it?
Either way works for me. So given the first half of your talk where you're talking about it's a very complicated multi-factor disease, is you're following epigenetics, showing that there is a validated target that you can use that will affect a substantial population? Is that your expectation? Or is the target going to be multiple pathways, multiple proteins?
Yeah, so I think there's a couple answers to the question. I mean, I think one, there's a lot of work to be done, and the answer is not a simple answer. I think on the I'll talk about on the non-immune and immune side. You know, on the immunological side, you know, I do think that there's some opportunity for novel targets. You know, there's clearly genes that are coming out of the genetic studies that are expressed in cell states that are expanded in some populations. Those can emerge as novel targets. I also think a very important, you know, big picture thing is the idea that there's a subset of individuals that have fibroblast predominant disease. There's a subset of individuals where, for whatever reason, their fibroblasts are really proliferating. And for them, what we see, targeting them with immunomodulatory, you know, hitting them with a T cell drug or a B cell drug doesn't get them better. So I think there's a really big unmet need in fibroblast biology. And obviously there's specific targets one can go after and specific modalities, but I think that whole kind of axis of tissue inflammation is largely been ignored. So so I think that's where there's a lot of potential. Diego?
Hi, great talk. But I wanted to ask a question about endogenous retroviral elements. I was wondering if you had quantified specifically HIVK, or also known as HML2, in these rheumatoid samples, as well as the chromatin accessibility at these elements, and seeing if you notice any patterns.
So it's going to be a short answer. I think the answer is no. We hadn't done that. I don't. Is that the kind of thing you can do with single cell data?
Yep, it's very recent. The algorithms only have really come out recently. And yes, you can actually get at locus specific expression. In my head, I'm wondering if patients actually have chromatin accessibility differences on top of these endogenous retroviral elements, which would explain a lot of their ability to generate autoimmunity.
Oh, that's great. Yeah. Yeah, you know, that's a great, you know, obviously I guess we have the data to look at this, so we should look at it. We haven't done it. But yeah, if you could drop me a line, I would love to.
Yeah, I mean, it's kind of, I'm asking because I've actually played around with a couple of blood data sets and actually have quantified these elements, and it works. It doesn't work great, but it kind of works, and there's some cool stuff. So definitely, I'll shoot you an email.
Yeah, perfect. Thank you, Diego. Other questions?
Other questions? If there are no questions, let's thank Soumya again for a wonderful talk. Anyway, thank you.
Thank you, everyone. Thank you for having me.
It's a pleasure. Take care. Bye.